Hi, I'm Jupinka, and this is our enzyme lab activity for Biology 200 Lab, Group D. My lab partners are Roya, Dixia, and Isabel. So in this lab, we we experimented with enzymes. Enzymes are protein catalysts that increase the rates of reactions, and they do this by speeding up reactions by lowering the activation rate needed. Enzymes are very important in humans, bacteria, and animals because they speed up reactions that are essential to life that normally wouldn't happen at a fast enough rate that we need for survival. The reaction was catalyzed by trypsin because trypsin breaks down azocasein, which was our substrate that we used. The role of trypsin in organisms is that it digests dietary proteins. So trypsin is found in the small intestine and helps digest dietary proteins. It's found in neutral pH levels in the small intestine. In our lab, we have a question and hypothesis. Our question is, how does the temperature affect the function of an enzyme? And our hypothesis is, if the sample is placed in a warmer temperature, it will have optimal enzyme activity. Our rationale for our hypothesis is that enzymes have an optimal performance in higher temperatures because they increase the activity of enzymes and the rate of reaction. The conditions of our experimental variable were temperature of 37, 50, and 60, and the conditions of our pH were level 9, and our conditions of salt were a concentration of 0.15 moles. So we had a lot of steps throughout our experiment. Started off with labeling all of our test tubes with different temperatures. We then added into the test tubes 450 milliliters of reaction buffer, 500 milliliters of azocasein, and 50 milliliters of trypsin. And then each tube was placed in a different temperature water bath of either 37 degrees Celsius, 50 degrees Celsius, or 60 degrees Celsius. The buffers were allowed 10 minutes to incubate, and then we took the test tubes out and added 500 milliliters of TCA. Then we centrifuged the tubes at max for 3 minutes. After 3 minutes, we transferred the supernatant into a clean cuvette and blanked the spectrophotometer at 440 nanometers using a cuvette with water. We then recorded the absorbances of our experimental groups once the spectrophotometer read 440 nanometers. So a really important part of our experiment was adding TCA. So we added the TCA to stop the reaction of the precipitation of macromolecule. So it was also very important to centrifuge the TCA, and the TCA was centrifuged because we had to separate the larger particles from the solution, and the pellet consisted of the enzyme and the substrate. So the substrate we used was azocasein, and sulfanilamide correlates to the ability of trypsin and the effects it has. So our experiment had two control conditions. The first one was our salt concentration, which we used at 0.15 moles, and our other condition was a pH of 9. And we used this pH because after seeing other groups that did their project on pH, we realized that that was the optimal temperature. So the negative controls, we prepared them the same way as our experimental groups, except instead of adding the enzyme of trypsin, we added water. In our experiment, we used 3 replicants, and we did this so we could reduce the error, find the outliers, and measure the variability. So based off of our activities, we can infer that the average of the 62 degrees Celsius was 0.209, and the variability was 0.011. For the 50 degrees Celsius, the average was 0.59, and variability was 0.088. For the 37 degrees, our average was 0.384, and our variability was 0.074. Okay. So our t-test results showed that the absorbance at 50 degrees Celsius and 37 degrees Celsius, as well as the absorbances at 50 degrees Celsius and 62 degrees Celsius, were significantly different. So the key results in variability of our experiment was that there was a significant difference in absorbances at a temperature between 50 degrees and 37 degrees Celsius, with a p-value of 0.036, and between 50 degrees Celsius and 62 degrees Celsius with a p-value of 0.001. Because a p-value is less than 0.05, the difference is significant, so that we can conclude from our results that the null hypothesis is supported by 5% confidence, and our alternative hypothesis is supported by 95% confidence. Our experimental design was adequate to test our hypothesis. However, our execution was not. We had errors mainly with maintaining the temperatures of our water baths and with our spectrophotometer. So our hypothesis was partially supported by the experiment because we did show that temperatures at 50 degrees Celsius is the temperature that trypsin has an optimal performance at. But we were incorrect because the highest temperature that we tested did not have optimal performance because it denatured it. Okay. Compared to other groups, we were the only group that tested temperature as our experimental value, but we used a pH level of 9 and a salt concentration of 0.15 moles, because that is what the other groups used in their experiments and proved that that was the optimal variables for trypsin activity. Yes, I think that our choice of control variable conditions resulted in the highest possible rate of enzyme activity. We maintained a pH level of 9 because the three other groups that tested pH, their experiments did prove that 9 had the optimal results because trypsin had a better performance in a higher pH rather than lower, and much higher than 9 showed to denature their enzymes. So a common source of variability that we had in our experiment was with the water bath. It was problematic because this resulted in all of our tests not being held at the same temperature, which caused our trials to fluctuate in temperatures and absorbances. Between our first and second experiment, we did have a few major changes and improvements. For example, we increased our pH level from 8 to 9 based off the observations of other groups. We also increased our experimental temperatures because our first time around we used more lower temperatures and didn't have the best results for that. So these changes allowed us to have more ideal conditions for our enzymes. And in future experiments, it would be beneficial to make improvements related to preventing errors. So finding a better way to test our samples in water baths, or just in general, decreasing any error that we used. So the relevance of our conclusion is that ideal conditions of bacteria are normally found in warmer temperatures because that is where they thrive. Warmer temperatures increase the rate of growth and helps bacteria maintain homeostasis in order to survive and grow compared to higher or lower extreme temperatures. And that is our lab.